negative earlier today, so I'm going to take my mask off. I hope that's okay with you, and I am fully vaccinated as well, so I think I'm not going to be transmitting viruses at you. At least I hope not. So, the origin of life. It's one of those questions when I started out in research that it seemed almost impossibly difficult to even frame the question. How can you possibly know? And it's been a field which has changed tremendously over the last 20 years or so. And biology has come to play a much greater role than it did. This has really been questioning chemistry until relatively recently. The book possibly that inspired me, and inspired quite a few people, this book, What is Life, by Schrodinger. And he kind of fails to answer the question in many respects. Virtually everybody fails to answer this question, what is life? If you want to understand the origin of life, we kind of want to start out by saying, well, what is life? And nobody can answer that. Because it's a process over time. There's a long continuum going from very simple prebiotic chemistry up to the complexity of genes and proteins and cells and ribosomes and things. It's a long, long distance, and I want to try and capture some of that distance this evening. So Schrodinger himself broke it down into two themes in his book. One of them was to do with genes and information, and he was one of the first people to talk in what I would describe as modern terms about code scripts. This was during the Second World War. Encryption was very much in the air, and Schrodinger was a physicist thinking about information. And so this is quite modern terminology, thinking about a code script. But he also talked about entropy. And here, to my mind, his language is much more opaque. He's talking about life feeding on negative entropy and continually sucking orderliness from its environment. And I don't really have a very clear idea of exactly what he has in mind when he talks about sucking orderliness. But he did have a footnote to the second edition where he said, if I'd been catering for physicists alone, I should have let the discussion turn on free energy instead. And as a biologist, that makes much more sense to me. So just so we're on the same page, what is free energy? Well, it's the energy available to power work, things like muscle contraction, but basically any enzyme activity and so on. It's all coming from cell respiration in our own case, and I'm sure you're all familiar with ATP as the universal energy currency. But the idea that there's something special about the bonds in ATP, that goes back to its discovery 70 years ago or so. There is nothing particularly special about the bonds in ATP, these three phosphates joined up. It's really, there's a disequilibrium of about 10 orders of magnitude between ADP and ATP. If you just left a bucket of ATP, it would slowly hydrolyze down to ADP and onwards to other things. So what life is doing is pushing the equilibrium way from equilibrium, 10 orders of magnitude out. How do we do that? By burning food in oxygen. And there's a very specific mechanism which is very strange and which is what I'm most interested in, which is how that's actually done. It's done by what's called membrane bioenergetics, so ion gradients, electrochemical ion gradients across membranes, and mostly proton gradients. A difference in proton concentration between the outside and the inside of cells or across membranes. And this is as universal as the genetic code itself across life, and yet is very rarely thought of in the context of the origin of life, just because it's so complex. We have very sophisticated molecular machines, proteins, which are pumping ions across the membrane, the ATP synthase, which is basically a rotating motor. None of them are prebiotic, that's very clear. And how they arose is quite opaque, and the assumption then has been, well, it must have been much later in evolution, it cannot possibly be prebiotic. But then you have this problem of, well, how does it arise? Why is it so universal? What's so good about that? The other issue here is that it's tended to make people look for something which is more believable. And what's more believable? Well, starting with cyanide, for example, or cyanoacetylene, or flammamide, using sources of energy like UV radiation or lightning. You'll all be familiar with the Miller-Urey experiment, I'm sure. Using wet-dry cycles and so on. So this is probably the dominant idea on the origin of life at the moment, is that life started in terrestrial geothermal systems. Something a bit like Yellowstone, but moved back 4 billion years to a different atmosphere where there wasn't any oxygen yet. The problem from my own point of view with this is it doesn't look much like life itself as I know it as a biochemist. Now, that doesn't mean it's wrong, it just doesn't join up very comfortably. And I think at the very least it suggests that we need to think about how do we join up prebiotic chemistry with cells and biochemistry as we know them. And basically, you can see this list, I won't go through the list, but down at the bottom here, there's problems with almost every aspect of this prebiotic chemistry. Beautiful chemistry being done over decades by some very, very smart people, but it just doesn't join up well with biochemistry. They would immediately criticize me back and say, yes, but there's a long, long distance going from prebiotic chemistry to cells that have genes and so on, and so we cannot possibly use life as a guide to its own origins. That distance is too great, and they certainly have a point. So can we use life as a guide to its own origins? There's a nice quote from Christian de Duve that I'd like to spend a moment on. He says, how did proto-metabolism come to be replaced by metabolism as we know it? So effectively prebiotic chemistry becoming biochemistry. The obvious answer to this question is that the appearance of catalysts, whether ribozymes, protein enzymes, or both, was responsible for the transition. So makes sense to me. We have to ask how catalysts with appropriate properties came to appear, and the only scientifically plausible explanation, he says, is that catalysts arose through selection. And I'm with him all the way on this. When he says scientifically plausible, I guess what he's saying, he was brought up as a Catholic, I guess what he's saying, if we assume that God didn't do it, then what else is there left? And really selection is about it. And here's the key point. Enzymes are selected only if they fit into protein metabolism. In other words, if you amplify or improve in some way things that are happening anyway, the kind of geochemical flux becomes a biochemical flux. Enzymes, catalysts, ribozymes, whatever it may be, are selected if they improve, if they enhance that flux in some way. In other words, it's working just as we understand natural selection. We can worry about what are the units of selection, how does this all work, but as a kind of underpinning principle, that makes sense to me. It still doesn't mean that we couldn't have followed a path that winds around all the place, but Occam's razor might say let's assume the shortest path from prebiotic chemistry to biochemistry, and let's see where it takes us. So how do we know, first of all, what was this prebiotic, well, what was the original biochemistry? Where are we trying to get to? Nothing like our own biochemistry necessarily, because we are aerobic organisms, we're multicellular, we're a long, long way from the origin of life. So what did the first cells look like? Well, this is a conceptual tree of life from Bill Martin, who was one of the really the leading thinkers in this field. And this is from 20 years ago now, and this was a very radical tree when it was put out, and it's still pretty radical even today. But 20 years ago, not many people would have believed it at all. And now, I would say, it's not exactly mainstream, but it's a lot closer to mainstream than it used to be. What we're seeing here is the eukaryotes up at the top. So where are all eukaryote cells that have got a nucleus with the DNA in the nucleus? What you can see here, this is this green line of chloroplasts, the acquisition of chloroplasts, endosymbiotic cyanobacteria giving rise to chloroplasts. And down here, we see a bacterial endosymbiont that goes on to become the mitochondria with an archaeal host cell. So what this tells us, this is now more or less mainstream. It may, we're not really absolutely in agreement about what's the host cell, how did this happen only once, did it happen more than once, when exactly. But the idea that there was some kind of relatively early event that gave rise to eukaryotes is now more or less universally agreed by people in the field. That means that eukaryotes are not relevant to the origin of life. They're a derived domain. They came from the bacteria and the archaea, and they've got nothing to do with the origin of life. So we can discard everything to do with our cells and eukaryotic biology from the equation. That makes things both easier and slightly more strange. The common ancestor then was the common ancestor of bacteria and archaea. This is Luca, the last universal common ancestor, and Bill has two separate emergences coming from what looks like a hydrothermal vent down at the bottom. He's not saying they arose independently, he's saying they shared a common ancestor, and that common ancestor was living in a specific environment. Why would he even say that kind of thing? Well, it's because phylogenetics and just genetics and biochemistry says there are some paradoxical differences between bacteria and archaea as domains. Plainly, they share a common ancestor. Things like the universal genetic code is shared across all of life. Ribosomes, the structure of ribosomes in bacteria and archaea are a little bit different, but they're basically superimposable almost. Transcription, translation, the genes involved in that are found in both bacteria and archaea, they're homologous. Some intermediary metabolism, bits and pieces of the Krebs cycle, glycolysis, things like that. Some of it is found in both domains. The chemistry itself is found in both domains, not always the genes. And membrane bioenergetics, which is my main theme in this lecture. That's also the use of proton gradients across membranes and enzymes like the ATP synthase is found in both bacteria and archaea. So what's different? The cell membrane is different. Not in the biophysics, its thickness, its proton conductance, that's pretty similar in bacteria and archaea, but the chemistry itself, even the stereochemistry, is quite different. I mean, just not homologous. Along with the enzymes that are responsible for lipid synthesis. Same with the cell wall, the chemistry is quite different in bacteria and archaea. So the boundaries between the inside of a cell and the outside world are just not remotely similar in bacteria and archaea in their chemical properties. Pathways like glycolysis, which you might think would be ancient and ancestral, the chemistry is conserved, but the genes themselves are divergent to the point that they appear to have nothing in common. DNA replication, I mean, this is crazy, but you know, there are several hundred genes involved in DNA replication, and only one or two of them appear to be homologous in bacteria and archaea. The rest are not. What does it tell us? I don't really know. It could be a completely separate origin, but whatever it tells us is that there's a problem here that we need to address. Respiratory chains, that are the pumps that are pumping protons or sodium ions or whatever it may be, they pass around bilateral gene transfer, but there is some suggestion, again, of independent origins in bacteria and archaea. So it's a bit of a paradoxical cell, Luca. And I like paradoxes. There's a lovely quote from Niels Bohr about paradoxes. He said, "How wonderful that we've met with a paradox, now we have some hope of making progress." I think it forces you to think as soon as you come across a paradox. And here's the paradox for me in this: membrane bioenergetics are universal across all of life, but the membranes themselves are not. Now the paradox, there's various different ways we might resolve it, but it could be resolved, most interestingly, if life originated in environments where there were geological proton gradients, it's geologically sustained proton gradients, across some kind of inorganic type barriers. And there is such a place. It was discovered by Deb Kelly, captain of the Alvin submersible back in the year 2000. These are alkaline hydrothermal vents. They're quite dissimilar to the black smokers that most people are familiar with. Black smokers are essentially volcanic. You have water interacting with magma at thousands of degrees, so the water is superheated, coming out at often 450 degrees. And you see this belching black smoke coming through a chimney, and you can see the chimney structure here. Lost City, that Deb Kelly had discovered, you can see that she's holding here a lump of Lost City, and you might be able to make out that it looks a bit like a mineralized sponge. There isn't a central chimney, and you can see again in the pictures, there isn't smoke belching out of the top. They're sometimes called white smokers, but often they don't smoke at all. And you can call them white non-smokers, I suppose. But these are actively venting systems. There are hydrothermal fluids coming out of them. It's just that they don't precipitate in the same way. Now, this kind of vent is probably really common. It's found unusually now. There are a few other examples known. But basically, it's formed by the interaction between rock and water. So rock, the submarine crust beneath the oceans, especially near the spreading centres, near, say, Mid-Atlantic Ridge, which is where Lost City was found, where the mantle is especially close to the surface. The mantle is made up of minerals like olivine, which are rich in magnesium and iron, and will react with water. And at higher temperatures and pressures down there, that reaction is pretty fast. Water can percolate down through cracks into the crust by distances 5-6 kilometers perhaps down into the crust, where they'll react with the olivine and metamorphose it into serpentine or serpentinite. And it's basically an oxidation of the ferrous iron in those rocks to ferric iron, ferric hydroxides. The key point is that it's releasing hydrogen gas in alkaline fluids, quite strongly alkaline fluids. And this kind of thing should be happening on basically any wet rocky planet or moon. And we know now, for example, Enceladus, I'm sure a few of you will be familiar with these plumes on Enceladus, which jet about up to 200 kilometers out into space from the surface, through the ice. The implication is there's a liquid ocean underneath the ice, and I think the ice is about 5 kilometers thick on Enceladus. And beneath that ocean, there must be hydrothermal systems. The reason I say that is these plumes are alkaline, they're rich in hydrogen gas, they're rich in some organics and simple things like methane. And their pH is around about 10. So it looks as if this is serpentinization going on beneath the liquid oceans on Enceladus. And the oceans themselves have become alkaline. Similar things seem to be happening on Europa, where the oceans are actually larger than the Earth's. And there's traces of methane occasionally possibly detected on Mars. Nobody can quite agree about that. It's more, if it really is methane on Mars, it's more likely to be serpentinization of trace amounts of water than it is to be methanogens, for example, producing it. We don't know. Hopefully we'll find out. But if there's anything there, it's more likely to be a geological process. Now these kind of vents were discovered, were hypothesized to exist 10 years before they were discovered by Mike Russell, who I think throughout the 1990s was a bit of a marginal figure in the field. He was putting these ideas forward through the 90s. Nobody really listened or believed him until the discovery of Lost City, and then they became quite mainstream. And this is a Photoshop job that Nature did in a feature article about Mike Russell in 2006, and they dressed him up as Erasmus, the Renaissance man, and called him Naissance Man, Naissance as in the birth of life, and they've got a bit of Lost City vent behind him, and this reactor that he built at JPL. And I'm showing also here a cross-section of a bit of Lost City. You can see again that it's this labyrinth of interconnected pores. It's really like a mineralized sponge. Now, this is mostly calcium carbonate, aragonite, and some brucite. But 4 billion years ago, before there was any oxygen in the atmosphere, when the oceans were much richer in iron and nickel and things like that, these should have been included catalytic iron-sulfur minerals and iron-nickel sulfur minerals, and that's what Mike Russell was proposing really in the late 80s, early 90s, that kind of period. The key points: high concentrations of hydrogen, that's true at Lost City, much more acidic early oceans. We don't know, it's hard to constrain, but there could have been up to a bar, if not more than 1 bar of CO2 in the early atmosphere, and that would have made the oceans probably pH 5 to 6, that kind of range, so a lot more acidic than today. So that means proton gradients, acidic early oceans percolating into these vents, alkaline fluids, pH 11 or thereabouts going in, and so we can have quite steep proton gradients across catalytic iron-sulfur walls. So this is the idea that there's an electrochemical flow reactor down, possibly the entire seafloor, serpentinizing in this way, producing this on any wet rocky planet. It's really a rather beautiful idea. It doesn't mean it's true, but it's a lovely place to start. And this is really where I got excited about it, because topologically, this is a bacterial cell, and we have a pump in a membrane, an organic membrane, 5 nanometers thick, and it's pumping out protons. I'm going to stick with protons as an example. And here we have a core in one of these hydrothermal vents, and we have alkaline hydrothermal fluids, and we have acidic ocean waters percolating in. So with a cell, we have a difference of equivalent to about 3 pH units, maybe 4 pH units, if we include both the concentration difference and the electrical charge, and convert the charge into pH units. It's about 3 to 4 pH units difference. Acid outside, alkaline inside. We have exactly the same thing here. Acid outside, alkaline inside, potentially 3 or 4 pH units difference across it. So both the magnitude and the polarity are equivalent to each other. Doesn't, again, mean anything beyond here's a starting point. Can we imagine any way to go from here to here? And that becomes then the domain of science, because if we can imagine it, we can begin to test it. I go back to Mitchell himself with this, who put these ideas forward in the early 1960s. He worked with Jennifer Moyle. She basically did all the experiments. I'm told that Mitchell himself was cack-handed in the lab. He was a genius thinker, a very awkward and difficult man by all accounts. Jennifer Moyle was a meticulous experimentalist. I don't think that she's had the credit that she deserved. Mitchell won the Nobel Prize by himself, I think today that would not happen, or at least I hope it would not happen. So without Jennifer Moyle's experiments, Mitchell's ideas would have been laughed out of house and nobody would have ever have believed him. But he was a genius thinker, and this is a really weird idea. There's a lovely quote from Leslie Orgel who said, "Not since Darwin has biology come up with an idea as counterintuitive as those of the great physicists, Einstein, Heisenberg, Schrodinger, and so on." It really is a strange thing. Why on earth would you have energy transduction instead of just being chemistry? Why do you have a membrane? Why do you have an electrical charge on a membrane? Why do it that way? It just seems odd. Now, Mitchell himself gave a talk at a conference in Moscow in 1957 on the origin of life, invited by Oparing, and various other kind of grandees of British science were there, J.D.S. Haldane and people like that. J.D. Bernal was there. Mostly they were communists. Mitchell himself was not at all a communist. He was at Cambridge in the 1940s and drove a silver Rolls Royce around Cambridge. And his father was, no, his uncle rather, was the founder of the Wimpey construction empire. So they built Heathrow, they built various airports and ports and houses after the war. So Mitchell was quite a wealthy man, and in the end founded the Glynn Research Institute in a country mansion that he bought with the proceeds of all of this. At this conference, though, in 1957, I love this quote. He said, "I cannot consider the organism," and here think about bacteria, not our cells, "cannot consider the organism without its environment. From a formal point of view, the two may be regarded as equivalent phases between which dynamic contact is maintained by the membranes that separate and link them." Virtually everyone who works on the origin of life says, here's a definition of life, and here's what's not alive, and I want to study life, because that's what I'm going to work on. And what Mitchell is saying here is I've got a membrane which is 5 nanometers thick, and on either side of it there are two phases which are equivalent, which are separated and linked by this membrane in the middle. And this phase is the inside of a cell, and this phase is the outside. And if you think about the origin of life, before there is really such a thing as cells or things that are alive, this idea of equivalent phases is really quite appealing. It makes a lot more sense. You're dealing with what's crossing this membrane and what effects does it have. So I find this a really helpful way to think about the question. And what Mitchell was thinking about in those early days was really bacterial physiology. He was wondering how is it that bacteria keep their inside different to the outside? How is it that these two phases are maintained as effectively separate phases? And he realized, it seems obvious to us, but he realized that the membrane proteins were going to have to work like enzymes. They're going to have to specifically recognize particular substrates, and they're going to have to pump them in or out, often against concentration gradients, so it's going to cost energy. So, you know, it seems hard to believe that that wasn't obvious to everybody at the time in the 1940s, but it was really Mitchell and co-workers at that time who were kind of crystallizing these ideas. So it costs energy to pump things out. And conversely, if you allow things to flow back in again, in principle you can conserve that energy and put it to use. And this was the leap from how do bacteria keep their insides different to the outside, to thinking about respiration in mitochondria in multicellular organisms and realizing that a gradient is needed and protons are involved and here's how it can possibly work. This is kind of a leap of the imagination. And so this is what's powering respiration, and this is what's powering bacterial cells. And I want to say it's powering work. The influx of protons can power work. ATP synthesis was what I thought about for a long time, and I'll touch back on this towards the end, but I never could think of any way in which an ATP synthase is a molecular machine. How could you have a prebiotic halfway house to that? CO2 fixation is what I've been thinking about over the last few years. It seems much more of a plausible and feasible way for prebiotic chemistry to be driven by proton gradients. Why would I say that? Well, the most ancient cells that we know about are methanogens and acetogens. Not everyone would agree with that statement, but there's quite a lot of evidence to suggest that these are ancestral groups within the archaea, the methanogens or archaea, and the bacteria, the acetogens of bacteria. And they both live from this reaction between CO2 and hydrogen. In the case of methanogens, to produce methane as a waste product, and in the case of acetogens, to produce acetate, acetic acid or vinegar as a waste product. And the delta G naught, so the free energy, is released. It's exothermic overall, exergonic overall, not strongly so, but enough to basically power growth. This is the acetyl-CoA pathway, and it's the only pathway of CO2 fixation that's found in both bacteria and archaea. There are differences, significant differences in the actual wiring of it in bacteria and archaea, but none of the other pathways of CO2 fixation, there are 6 of them in all, are found in both bacteria and archaea. So this looks to be one of the most ancient, if not ancestral. And if we think about core biochemistry, CO2 and hydrogen just keeps on coming and coming and coming and coming. So we're starting with CO2 up here, and in brown, I hope you can make it out, is H2, 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 H2. Lots of hydrogen involved. In green is CO2. We've got CO2, CO2, CO2. It's, you know, step after step is basically hydrogen and CO2. What are we doing here? We're starting with CO2. We're coming through acetyl-CoA. Forget the CoA. Just think thioesters, prebiotic thioesters. And we're coming into the Krebs cycle, basically, the reverse Krebs cycle, pyruvate, oxaloacetate, alpha-ketoglutarate. We go from pyruvate to glyceraldehyde into sugars, to ribose, for example. We go to amino acids, oxaloacetate to aspartate, and so on, alpha-ketoglutarate to glutamate. The blue arrows show prebiotic chemistry that's been done in the lab, not necessarily always under the same conditions, and there's a lot more work needs doing here. But 5 years ago, really none of that was true, or none of that had been demonstrated. Now, most of this has been demonstrated, not to the level that I would like to see it done, but to a level which makes it believable that it really can happen. And in the red boxes here, these are the precursors for nucleotide synthesis. If we want to get to DNA, RNA, DNA, to information and so on, and we want to use biology as a guide and follow the steps that biology is doing, this is the starting point, this is intermediary metabolism, and here's where we start to the precursors for nucleotide synthesis. So if we want anyone to take these ideas seriously, then we need to demonstrate that it's possible to do these chemical syntheses starting from these precursors and going to nucleotides. And I'll come on to that in a little while. So I've said CO2 and hydrogen really central to metabolism, and we should know that because theoretically thermodynamics have been saying for a long time that if you start in alkaline conditions with alkaline hydrothermal conditions, 50, 75 degrees centigrade, starting with CO2 and hydrogen, we should get it's exergonic, exothermic to produce total cell biomass, quite strongly so. Especially for amino acids and fatty acids. Nucleotides you can see are up here. It's endergonic to produce nucleotides. But if you can couple some of the energy released down here to nucleotide synthesis, then overall we should get total cell biomass. I still find this difficult to get my head around. Basically, if you've got a Falcon tube and you've got a mixture of hydrogen and CO2 in it at 50 degrees centigrade in alkaline conditions, and you shake it for a little while, you should get cells dropping out. This thermodynamics says you get cells. Cells are more stable than a mixture of hydrogen and CO2. That's gobsmacking. But of course it doesn't happen. Why doesn't it happen? It doesn't happen because there's a barrier. There's a thermodynamic barrier to get through the first 2 steps. To get to formaldehyde up here, and then it's downhill all the way to methane. This is the redox state of most of intermediary metabolism. So in terms of energetics, it's slightly downhill to get there, but not that much. So here's the barrier. This is why if you shake CO2 and hydrogen together, you don't get cells dropping out. You've got to somehow lower this barrier. And the way that methanogens do it is by using a proton gradient across the membrane. And this, to me, is really the single key thing that makes all of this plausible and feasible. How do they do it? They use a membrane protein called the energy converting hydrogenase, ECH, and that takes the electrons from hydrogen and pushes them onto ferredoxin to form reduced ferredoxin. This is an iron-sulfur protein. This is also an iron-sulfur protein, and ferredoxin reduces CO2 to make organics. Let's just keep it simple. And now it should not happen. It requires this proton gradient to allow it to happen. Let me come this side for a little while. So here's the problem. Hydrogen, under the conditions under which methanogens grow, is just not reducing enough. It's got a reduction potential of about -300 millivolts. And the more negative it is, the more reducing that is. So minus 300 is not particularly reducing. Paradoxin has got minus 500 millivolts under the conditions in these methanogens. So there's a 200 millivolt gap. There's no way that hydrogen can reduce paradoxin. It just should never happen. But it does happen because we have this proton gradient coming right through a pore in the membrane onto these FES clusters here. Exactly how that works, I won't talk about it now. I can come on to it if people want to ask questions. I have some ideas, but the simplest way of thinking about it is to realize the involvement of protons. So if this is, again, just think uphill here, it's uphill to go from hydrogen to reducing CO2 to formaldehyde. So this is, again, reduction potential, but we don't need to worry about the details, it's uphill. So at pH 7 or any other pH, hydrogen cannot reduce CO2 to formaldehyde. But in a hydrothermal vent, we have hydrogen at pH 11 and we have CO2 at pH 6, say. And that means in principle it should happen. Why? Because for hydrogen to push its electrons onto something else, what's left behind? Protons. The protons in alkaline conditions will react with hydroxide ions immediately. It's an acid-base titration. It will fizz, and you will produce water in water. This is another theme that we could come back to later on. And so it's encouraged under alkaline conditions. Whereas CO2, if that picks up an electron, then it's picked up a charge. It's not going to pick up another negative charge very easily, but if it's in an acidic environment, it picks up a proton, balances the charges, takes another electron, another proton, it can keep balancing the charges. So it's easier to reduce CO2. This is, for electrochemists, just the Nernst equation. It's 60 millivolts per pH unit if protons are involved in that reaction. So the redox potential of both hydrogen and CO2 and some FES clusters depends on pH in that way. So in principle, something like this could happen. We can have protons crossing this barrier, CO2 crossing the barrier, and sharp pH differences driving the reaction between hydrogen and CO2 to produce, not formaldehyde specifically in this case, but organics generally. On this side we have the alkaline fluids. And all of this is happening inside these micropores. The outside is not the ocean, the outside is another of these pores. So it's all happening inside these vents. That's the idea, and if you allow it to its logical conclusion, then we end up producing protocells of some sort, initially made from simple monomers, fatty acids, for example, which, again, we can imagine. We have to imagine a lot because there's really very little that we actually know. We have to imagine, and then we can try and test. And I imagine that we could have smaller nanocrystals of iron sulfide collated by organic molecules like amino acids, which bind to the membrane and which drive this. And now we have an organic membrane here, and we've got the same topology. We've basically got a continuity between geochemistry and biochemistry driven by the same gradients. It's all imagination. I've been trying to test these ideas for quite a long time. It's great fun. It hardly ever works. These reactors were somewhat crazy. These were producing small vent systems inside the reactors. We've tried having some kind of mineralized sponge in there to simulate the currents themselves. We've ended up with microfluidic reactors, which are just a lot easier to repeat. We've had some success, but I've hardly published any of this particular. We made formaldehyde, for example, but we couldn't repeat our own experiments because the reactor itself was basically so stochastic. We've had enough to make me think it's not a completely stupid waste of time, but neither have we ever nailed it. It was actually a former student of mine who went on to build his own microfluidic chip, and he was kind enough to include me as an author on the paper, but I actually had nothing to do with it beyond being thinking of some of the ideas. This was in PNAS about a year ago, and what you're seeing here, again, it's basically the same idea. We've got hydrogen in alkaline fluids on one side, we've got CO2 and acidic fluids on the other side, and the hydrogen passes its electrons onto the barrier, which cross the barrier. This is one way of reading it, and I'll say more about that. Over here, there are various other possibilities to explain what exactly is crossing that barrier. But what Victor and Ruben Hudson showed using NMR, 13C NMR, and proton NMR, and using deuterated water and using 13C, he was able to show that the CO2 that became reduced was definitely the CO2 that was added to reduce through to formate, that the electrons came from hydrogen, that you needed a barrier for that to happen, that you needed a pH gradient. If you had a lower pH gradient or no pH gradient, it didn't happen. If you didn't have pressure, very limited pressure, only about 1.5 bars pressure, but if you lowered it down to atmospheric pressure, it didn't work either. If you don't have any hydrogen, it doesn't work, so that's down here. There are various controls that simply didn't work. So this is really, you might say, a trivial proof of principle, but it demonstrates that it is possible to drive CO2 reduction by hydrogen using pH gradients across a barrier. That's the first time anybody's ever done it. The experiment amazingly lasted for 6 seconds because the pressure was pushing things through their chip. We're trying to do it better. There's another former student of mine who's making some progress with pressurized chips as well. But it's a start. It's better than it was a year ago. We've also been wondering in these microfluidic chips, what really is crossing that barrier? Is it electrons? Is it protons going the other way? What's happening? And this is just showing that we have with laminar flow, without a barrier of any description, you still have pH gradients. This is just using a universal indicator. This is acid on this side, alkaline on that side, and a little bit of mixing in the middle. But when you have a precipitate, sometimes the precipitates form these very diffuse structures with plenty of water trapped in the middle. And we notice that they're always acidic, and here as well. We never saw alkaline fluids trapped in them, we only saw acidic fluids trapped in them. And so we looked into it in more detail and we did some stop flow experiments, and what we found is that basically protons are crossing this barrier about 2 million times faster than hydroxide ions going the other way. This was from the stop flow experiments. That means back of the envelope calculation suggests that we can have proton gradients as steep as 3 or 4 pH units across single nanocrystals 25 nanometers in diameter. That's a calculation rather than a demonstration. But we can certainly have, this is the distance, I think, let's see if I can see it, of about 70 micrometers between here and here. So these are not very good pictures really, but they demonstrate again a principle that we can have steep proton gradients in these systems. Which means at least this kind of thing that I showed you already is feasible in principle. So we need to go about testing it properly. And already there's several, you may think, serious problems here. What I'm showing you is a protocell with presumably mixed amphiphiles from bilayer membrane, sitting in a hydrothermal vent with acid on this side, alkali on this side, acidic ocean conditions, lots of divalent cations and so on. This is a nasty environment for protocells to be in. Can they really survive there? So we've been looking into that. The pH range they need to cope with will be 5 up to 12. The temperature range, maybe 50 to 100 degrees centigrade. Salinity, best guess would be modern ocean salinity. And plenty of divalent cations, magnesium, calcium ions. Again, the best guess is modern ocean levels of those. It's very, very difficult to constrain what was the salinity of the early oceans. Which amphiphiles should we even use? This is work from 20 years ago from Tom McCollum, doing a kind of hydrothermal Fischer-Tropsch type synthesis. And what that involves is basically, it was developed, in fact, in the 1930s by Fischer and Tropsch in Germany before the war to try and convert coal. Germany was rich in coal but had very little petrol, gasoline. And this was a method of effectively releasing carbon monoxide from coal and converting it into long-chain hydrocarbons. And it can be done under hydrothermal conditions as well. And here are the hydrocarbons that are being formed. And you can see that this is 15, 16, 10, 11. These are the chain length, the number of carbons in these chains. And it includes alkanols and alkanoic acids, that's to say. So starting with formate, at relatively high temperatures, 150 degrees if I remember rightly, where you have this gas-water shift. The idea is it will break down to release CO2 and hydrogen that would then react with each other to form the long chains. Interestingly, it only worked in steel reactors. They didn't see any of this in glass reactors. Why steel? One possible explanation is that in steel you have carbon bound directly to iron, so carbide bonds. And there are some carbide bonds in biology as well. In the nitrogenase enzyme, for example, is a carbide bond. So it may well be that the structure of steel is a catalyst really rather equivalent to some enzymes still involved in doing these processes today. So I wouldn't take that as a bad sign that it only works when you have the right catalyst there. It's interesting that the catalyst was steel. So we took those amphiphiles and we mixed them together and looked to see if they would form protocells under alkaline hydrothermal conditions. And this is what most people do. They use a single decanoic acid, a single 10-carbon fatty acid. And the steep bit of the curve here is where you get vesicles, where you get a bilayer membrane surrounding an aqueous space. Down here you get micelles, where there's no aqueous space in the middle. The fatty acid tails are all interacting directly with each other. And up here, everything's protonated and you end up with kind of fatty globules. This is optical density, so we're really looking at how much the passage of light is obstructed by the size of the things in it. Very little obstruction from micelles, much more from fatty globules, and vesicles are somewhere in the middle. This means that they survive at pH 7 and nowhere else. That's what the literature says, and we confirmed that. But here, if we have 6 different fatty acids in the mixture, you can see that it's a much more gentle curve, and we're getting vesicles up to about pH 9. And here, with a 5-to-1 mixture of fatty acids and fatty alcohols, we're up to about pH 10, where we're supposedly seeing vesicles. And then this was a 1-to-1 mixture, and this was a complete mess, and we thought, what on earth is going on here? But if you take it at face value, it implies that vesicles are absolutely everywhere, because these values are intermediate, they're in this kind of range. And that's what we see. This is cryo-EM of bilayer membrane vesicles at pH 12, 70 degrees centigrade, and basically modern ocean salinity with graduated cations. And this is pretty much the same thing here with confocal microscopy, and again, you can see there's plenty of vesicles there. These conditions, if anything, favour the formation of vesicles, so long as you've got this messy mixture of different amphiphiles, things that deprotonate or reprotonate at different pHs. It's that allows you to keep a membrane. And we were also interested to see, can we produce at least smaller nanocrystals of iron sulfides if we collate with amino acids like cysteine, for example. We were quite surprised to find not only does it work, but it works much better than I would have imagined. This is UV vis spectroscopy, and what we're seeing here is a shoulder, which is the diagnostic shoulder, if you like, for the 4Fe, 4S clusters, exactly the same clusters that you see in ferredoxin and energy-converting hydrogenase and so on. And it's concentration dependent, depending on the concentration of cysteine and iron and so on. This was published a year or so, a couple of years ago. And we've showed as well with Mossbauer spectroscopy, these pick up the different spin states of the different types of cluster. And again, it's concentration dependent. Higher concentrations of cysteine, we're more likely to get 2Fe, 2S clusters, and mononuclear clusters with a single ion chelated by cysteines. Whereas up here we have a more interesting mixture, including the 4Fe, 4S clusters. We're only getting about 10-15% 4Fe, 4S clusters in this mixture, but they're there all right. And they're redox active as well. This is cyclic voltammetry. And they're not as reducing as I would like them to be, but we know that this is a composite mixture, and so we need to kind of break down that and work out the redox properties for the individual clusters. Other things we've been doing, I said these are the starting points for nucleotide synthesis. We've not got that far yet, but we've been looking at pyrimidine synthesis. This is uracil. This is the nuclear base, not the nucleotide itself. And this is the pathway starting from biology of ammonia and columnate and aspartate. And these are the steps going through to uracil. And in the red box here, this was some steps that we optimized. So this is going from dihydroorotate through to uracil by orotate. And what you probably won't be able to make out, well, these are different metal ions catalyzing the step to orotate. And this is molybdenum here. We're getting a moderate yield of orotate. So really just this step. But here is uracil in the presence of copper 2+, but nothing else really. We're getting quite a good yield of uracil. When we optimize the conditions and we consider a range of pH, a range of temperatures, a range of pressures, a range of salinities and so on, and then optimize for the best conditions, this is orotate and this is uracil, and this is uracil under the optimized conditions. Whoops. And those optimized conditions were 90 degrees centigrade, alkaline pH, salinity quite close to modern ocean salinity, and high pressure. So very consistent at the very least with the alkaline hydrothermal conditions. And this was a one-part synthesis of uracil, of orotate and uracil, and the best we got was when we switched pH from the acidic ocean type pH of 5.5 to an alkaline vent type pH of 9.5, and then we got the best yield of orotate. So this is really promising first steps. The synthetic chemists who've been starting with cyanide have gone way beyond what I'm showing here. But on the other hand, they've always said it's impossible to do this, and that's why they don't bother. So we are beginning to show that it is possible to follow the pathway of biology to synthesize the building blocks of life. So it is really possible to start with the right substrates. And some lovely work from a PhD student, Silvana Pinner, who's just finishing now. And this is the phosphorylation of ADP to ATP in water. And it's an interesting question. How do you do that if you don't have an ATP synthase or you don't have a pathway like glycolysis? And the obvious way of doing it is substrate level phosphorylation using acetyl phosphate, which is still used today in Bacterial and Archaea and still used to phosphorylate ATP to ATP. And this is showing with this is with acetyl phosphate, 30 degrees centigrade, pH 5.5 to 6. This is with acetyl phosphate, this is with carbon oil phosphate, and these are a whole bunch of other different possible phosphorylating agents down there. Nothing else works. Acetyl phosphate gives a yield of 15 to 20%. It doesn't work so well at pH 7, and it doesn't work at all at pH 9. And at 50 degrees centigrade it's faster, but it hydrolyzes faster too, and again it's pH dependent. Well, it's other people back in the 90s had shown that it's possible to use acetyl phosphate to phosphorylate ADP, so that's not original. But nobody else had demonstrated that only acetyl phosphate really does it. And then it only works in the presence of ferric iron. It doesn't work with, we tried a whole battery of other metal ions, and none of them work apart from ferric iron. Ferric iron is the only catalyst that will do this. Oops, it keeps jumping forward. And then the other really interesting thing is it works with ATP, but it doesn't work with the other bases. In other words, why is ATP the universal energy currency? Because it works with ATP and it doesn't work with GTP or UTP or any of the others. This is a peak with ATP here, and this, it looks as if there's something, but actually at naught hours we get the same. It's a contamination from the manufacturer in some of these cases. So we're getting no yield of the triphosphate in any of the cases apart from ATP. So it really looks as if ATP is the universal energy currency, because this, in water, this phosphorylation step using acetyl phosphate, which still happens, was favoured by prebiotic chemistry right at the beginning. The other thing which is interesting to think about is it works at acid pH but not alkaline pH, and you may think that that says, a problem with hydrothermal vents here. Actually, it's the opposite if you think about it. What do we want ATP to do? We want it to drive work. In other words, we want it to be out of equilibrium. We want it to be hydrolyzed to phosphorylate things. So how do we get it out of equilibrium if we don't have the machinery for doing that? Well, it will be synthesized in a membrane-type environment as acidic pH, and then it passes into more alkaline conditions where it's now out of equilibrium. It now doesn't want to form at all, and a quick calculation shows that's around about a 400-fold difference in terms of the equilibrium. So we're pushing it 2 or 3 orders of magnitude out of equilibrium just by the pH shift going from the membrane into the cytosol, if you like. So it's a useful way of thinking about how these things get going. It's not enough power yet, but it's at least a start. So for the last 5 minutes, let's just check that I have 5 minutes. Okay. Can you give me 5 minutes? No answer. So I'll take that as a yes. I want to talk about the origin of information in biology. This is a teaser, really, as much as anything else, because these ideas are not fully developed yet, but they're a nice way of seeing the question. So this is modelling, mostly, and this is Raquel Nunes Palmero, the PhD student. This is the kind of thing you've seen already, a protocell with CO2 fixation going on across the membrane to produce something equivalent to acetyl-CoA, so an activated acetate. And we're assuming branching metabolism, so going to fatty acids, going to some energy currency like acetyl phosphate, to sugars, to amino acids of different types. And we're assuming some kind of positive feedback. So if this is cysteine, binding to iron and sulfide, forming 4Fe4S clusters, is driving CO2 fixation. And so we've got a positive feedback which is going to enhance cell growth. And then here, same kind of thing, but we're now going to go further. We're going to go as far as nucleotides. We're taking an energy current, so we're taking the sugar phosphates, we're taking amino acids, and we end up with nucleotides, and we're assuming then some kind of positive feedback. This time on CO2 fixation, there's grounds for that. Think of these as naked cofactors, and some cofactors, folates, for example, and molybdenums, are derived from GTP. And they are involved in CO2 fixation in the thanogens and acetogens. So this is a perfectly reasonable hypothesis to assume there will be that feedback. But we can also consider all kinds of other possible feedbacks on all these other pathways as well, and we can assume also autocatalysis of nucleotide synthesis, which also, to make purines, you need the 6 phosphorylation steps to make adenosine or to make guanine, and those steps require ATP today. So there has to be some kind of positive feedback there as well, some autocatalytic loop on purine synthesis. What do we see? Well, it depends on how good the catalysis is. If we have weak catalysis of CO2 fixation by nucleotides, in this case, we get no increase in growth rates. But if that catalysis improves, then we see an exponential lift-off in growth rates. So we don't know how well they would catalyze things, but we do know that we see this dynamic that if they are decent catalysts, then we will certainly get some kind of lift-off. This is perhaps more interesting, is if you're having the feedbacks on individual pathways, you get very little benefit, and often it collapses, growth can collapse completely. And the issue here is that if you put too many resources down one pathway, then fewer go down other pathways, and you kind of unbalance metabolism, you unbalance growth. What we really need are general purpose catalysts, and actually nucleotide cofactors are general purpose catalysts. They're not like enzymes that catalyze very specific reactions. NADH will always pass hydrogen past 2H onto other things. It will always do that same job, so it's very general purpose. ATP similarly. And here, this is looking at growth rates, depending on catalysis of CO2 fixation on the y-axis, or autocatalysis of nucleotide synthesis on the x-axis, and you can see this is the region of fastest growth, where it's doing a bit of both, which is again similar to biology. Whereas what we've been hoping is that we will be able to find a set of conditions that would concentrate nucleotides, making it more likely that they would now start to polymerize. And what we actually found is that the only way to concentrate nucleotides is if they catalyze their own formation but don't have much of an effect on CO2 fixation. Because if they catalyze CO2 fixation, the cell grows faster, divides in two, and halves the concentration immediately. It's obvious when you think about it, but it hadn't occurred to us. And so, basically, if you have cells that are going to accumulate nucleotides, they will grow slower than cells that don't. So it's a useful way of, bringing you back down to earth and realizing that we need to think of other ways of polymerizing nucleotides other than just having a high concentration. So the last couple of minutes. The code within the codons. Where does information come from in biology? Well, there are patterns, and a lot of these patterns have been known about for a long time, but not necessarily from a strictly autotrophic viewpoint of the origins of metabolism. So this is what you've seen before. This is basically organized, starting with CO2, going into pyruvate and Krebs cycle intermediates and so on. The first letter of the codon is color-coded here. It's a G if it's grey, and this has been known about since the 1980s. That if the first letter of the codon is a G, then it's a one-step reductive amination from a carboxylic acid. So it basically correlates with the precursor of the amino acid that it goes for. And if it's a yellow here, it's an A at the first letter of the codon, and this is basically an extra step, very often involving ATP. Maybe it's a couple of extra steps, but basically you can begin to see the spatial patterning. So in green is uracil, so this is the most distance, and in blue is cytosine. So again, and this is the least number of steps from CO2 fixation. You can see quite a strong correlation there. This is cysteine here, which is a bit of an outlier. I'm not going to talk about that because I don't understand it. But we see this strong correlation. And over here, this is a little complicated to explain, but this box is where G is at the first position and A is at the first position, and this is now the second position. So with this grey, the second position is an A or a G with the yellow, if I remember rightly. Yeah, so G is with the yellow here. So this is now the second position, and what we're seeing is a pretty strong correlation in hydrophobicity between the second position of the anticodon, the base at the second position of the anticodon, and the amino acid it codes for. So in this case, A is the most hydrophobic of the bases, and valine is the most hydrophobic amino acid. This is from about 41 different scales of hydrophobicity. No one can agree about really what hydrophobicity is, how do you measure it. And so this takes all the scales, and you can see the violin plots. There's a strong correlation in these first two, where there's a purine at the first position, we see a strong correlation, but if it's a pyrimidine at the first position, we don't see that correlation. So these are, all I really want to get across is there are strong patterns in the code which suggests it's not random. This is not a frozen accident, as Crick once argued. And this is the third position, which most people, including me, would have said is purely random, that it can be almost anything at the third position. But this is an inverted codon table. This is a student, Stuart Harrison, who's also been doing the uracil synthesis that I talked about earlier, and he is a very clever thinker about biochemistry. And this is, if you've got a G at the first position, an A at the second position, and a purine at the third position, compared to exactly the same thing, G at the first position, A at the second position, and a pyrimidine at the third position, then we can predict which of these amino acids is going to be glutamate or aspartate, depending on the size. There are spatial steric hindrances, so both hydrophobicity and shape matter. It implies that there are direct biophysical interactions between amino acids and early RNAs, and this is the final slide then. What does that mean? I think this explains where information comes into biology. So we have far from equilibrium environment driving protocell growth from earlier on, protein metabolism following these favoured thermodynamic pathways, which ultimately leads up to nucleotide synthesis. I've not proved all of that yet, but I hope I've given you a sense that at least some of it works. In principle, in the modelling, we have positive feedbacks from nucleotides, for example, nucleotide naked cofactors on CO2 fixation. And that can, again, promote faster rates of growth of protocells, non-hereditary protocells, but positive feedbacks can improve their growth. This kind of heredity is not evolvable, but it can gain in complexity as you break down kinetic barriers to particular steps happening. As soon as we've got ATP synthesis, we have some prospect of polymerizing things, polymerizing nucleotides into short, random RNA strands within growing protocells. And if we have biophysical interactions between amino acids and these RNA sequences, the RNA sequences can be random, but they're going to template amino acids with to produce particular peptide sequences. And those peptides will depend on their function. Hydrophobicity, for example, hydrophobic peptides would tend to go to the membrane, and would tend to have membrane-type properties, for example, CO2 fixation. Whereas hydrophilic peptides are more likely to interact with ions, like magnesium, and take on hydrophilic-type properties like the RNA polymerase. And so we get function emerging from random sequences of RNA, so long as you have a protocell environment, so long as you have growth, so long as you've got a lot of biology going on that was already there from beforehand. And so these sequences, in principle, they're modeling this at the moment, but in principle they can optimize protocell growth from the outset, and so information has meaning which is acquired with function from the very beginning, and we do not need to look for some unknown law of physics to explain the emergence of information in biology. All of this really depends on my group, who are pretty brave to work on these kind of questions. I think this is what scientists should be doing. I would say that, wouldn't I? But they've been doing really beautiful work, and I'm proud to be associated with them, and I really thank them more than anyone else. I have to thank the funders as well, and thank you for listening. I'm very happy to take questions.
Thank you, Professor Lane. If there's any questions, can you just raise your hand? add H2 or CO2 supermolecules to build up like amino acids at the end. I wonder if there was any thoughts on how you select the correct amino acids.
It's not something I've given a great deal of thought to. I mean, it's something I tend to associate with polymers, which is to say, as soon as you have enzymes, then you're going to have necessarily steric chemistry for the standard kind of hand and glove type mechanism. So as soon as we've got real selection going on real peptides, then for sure we have to have stereochemistry in force at that point. Could it arise earlier? Yes, it could in these kind of environments, because mineral catalysts and so on may tend to produce one isomer over the other one. I'd say the evidence for that's weakish. It does happen, but I don't really see a need to insist on it earlier than a kind of polymer stage of evolution. So I don't have a strong view. I know there are people who have argued you know, cosmic chemistry can produce specific isomers. I don't doubt that it does, but I don't know that it's needed for the origins of biology.
Could life still be forming now in the hydrocline?
If the ideas that I've talked about are remotely true, then no, because the oceans now are oxygenated, the chemistry is completely different, you don't have much iron in modern oceans, and they're full of life as well, because you can basically eat anything that's there. So I would say the chances of life forming on planet Earth now is basically zero. What are the chances of it forming back 4 billion years ago in the Hadean is really difficult to know. My feeling is there's no reason to think it's so improbable. I would like to think that this kind of chemistry is facilitated on any wet rocky planet, and that the first steps, I think there's a pyramid of difficulty. Getting to things like ribosomes seems enormously difficult, which would tend to imply that you better make the first steps as easy as possible so that you're producing enough of the substrate. And a lot of the chemists would say, wow, well, that's why you need cyanide, you need to make sure this chemistry works really well. And they have a point. And I think at the moment we've got a long way to go to demonstrate that starting with CO2 and hydrogen produces enough of the precursors for biochemistry. You know, it's a very simple, specific challenge. Do it. And if after another 10 years I'm struggling to make formate out of these systems, then I think at that point I may throw in my towel and say, well, I'm not sure if this really does work. But at the moment, it looks as if it looks quite exciting. It looks as if doing the basic chemistry will turn out to be easier than we thought when we find the sweet spot.
Do we have any other questions?
Thank you for the talk. First question, sort of less serious. Are they holding molecules that correspond to the Krebs cycle in there on your slide?
Sorry, what was that about the Krebs cycle?
Are the members of your lab holding molecules
Yes,
that correspond to the
absolutely
Krebs
they are.
cycle?
Yes.
That's brilliant. But
It was actually a birthday present from a year ago, which was very nice of them. And I've just finished writing a book on the Krebs cycle, which is why they're taking the piss.
That's brilliant. But second, more serious question. So you've talked about the stability of the sort of protocells with lipids. Archaea, I think, have ethers as their membrane molecule. Is there any research going on into if life could have arisen as sort of with the ethers instead of fatty acids? And so if that influences the stability and other stuff like that?
Yes, I mean, we've done some work on this ourselves, making protocells. We added isoprenoids, so geraniol, which is a 10-carbon isoprene, again, without the head groups. I mean, and I said this in an earlier slide, but the difference between the bacterial lipids and the archaeolipids is a really deep divide and really interesting. It's the stereochemistry of the head group, which is particularly distinct. So archaea have a glycerol 1-phosphate head group, the bacteria have a glycerol 3-phosphate head group. There are fatty acids in some archaea, and there are isoprenes, cholesterol is one, for example, in bacteria and eukaryotes. So that difference is more of a kind of probabilistic difference rather than an absolute difference. But the glycerol 1, glycerol 3-phosphate, I'm not sure that there are any known examples. There may be one now. but it's pretty rare. And it begs this question, why is there such a deep gulf? And it may be partly linked now to membrane proteins that have been optimized to specific lipid backgrounds that they're in, so it may be quite difficult now to change your lipid context. But what about the origin? Why did this come about? Isoprenes, when you add them, have some unusual effects. They don't form bilayer vesicles very readily themselves. And if you add them to fatty acid bilayers, you need to add about 30 times as much geraniol compared to decanol, for example, as just a long-chain fatty alcohol. Why, I don't really know, but it seems to be to do with the packing in one way or another. And the other thing, when you add geraniol, they no longer stick to mineral surfaces. They don't absorb onto mineral surfaces. They tend to prefer to just be an isolated round protocell by itself. So just adding in isoprenes has a surprising effect. I don't know exactly why. It may be the cross-links. It may be there. I'm not really sure what the reasons are. The postdoc who was working on that is going to continue with it. He's now gone to Lisbon, and I'll look forward to seeing what he does on it. So my own feeling is the divergence between bacteria and archaea, probably there were mixtures earlier on of single chain amphiphyrals, and the real divergence was linked to the head group being added, a glycerol phosphate head group, which was done by enzymes using stereochemistry at a later stage with that divergence. And the key point, why would it have to be like that? If you want to tap a natural proton gradient and you do not have pumps, you've got to have a leaky membrane, leaky to protons. Otherwise things come in and can't leave again. You've effectively got a closed system. You need an open system. You need protons to come in and then leave again, and you need mixing. Otherwise, you have charge accumulating and gluing up everything. So you need early on, I would say, fatty acid membranes, maybe with some isoprenes, and only later do you want to seal them off when you can pump, and that requires genes.
Thank you.
I think we have time for like 2 more last questions.
If I got you right, you say that the first enzymes kind of have to be then involved in hydrogen transport and into this primary kind of metabolism. Are there then still molecular fossil enzymes around in organisms that live in those kind of areas?
Yes, it's hard to agree on what a fossil is in terms of an enzyme. I've mentioned the energy converting hydrogenase. It's an iron-sulfur membrane-bound protein that's found in these most ancient cells, but it's also in your mitochondria. It's the core subunits of complex 1 in the respiratory chain. So if that's not a molecular fossil, then I don't know what is. Interesting questions. Why do we have all these iron-sulfur clusters in our respiratory chains? There's 11 of them in complex 1. It seems to me to be reproducing an ancestral environment in one way or another to do with iron-sulfur clusters. And I don't know why so many. I think it's to do with modulating redox potential, to do with quantum tunneling and the distances between these centers and the probability of electrons being here rather than there. You can modulate that depending on how many of them that you have. You would lower the probability of it being on any one center if you have more centers. So exactly how that works, I'm not sure. But I think quantum biology comes into that kind of thing. The other kind of fossil enzymes, I would say the cofactors themselves, and people have been saying that cofactors are a remnant of the RNA world for a long time. And you can see them that way, and they are very often dinucleotide cofactors, but not always. The folates that I mentioned, they're not dinucleotides, it's derived from GTP alone. But you could also see it the other way around, that before you get to an RNA world, the first steps are to have single nucleotides or dinucleotides. Before you've really mastered the art of polymerization, you've got some catalysis from naked cofactors, which are doing the same jobs now, and they are later, they gain a peptide context. How do they gain that context? You don't need genes for it, it's just biophysical interactions between peptides that have the right kind of amino acids there, and the nucleotides themselves. So if you wish to bind a purine base, you better have hydrophobic amino acids close by, and so on. So I think we can go a long way with biophysical interactions and this idea that very often the active site of enzymes, especially if there's a cofactor involved, is conditioned by the biophysics.
Any last question?
Possibly a little follow-up.
Yeah, sure.
Are there then also secondary structure or kind of generally structural elements involved in those folds that you can kind of track down as being more basic than they would be in modern enzymes?
Yes, I mean, I've not thought much about that, but my guess is that you start out with short peptides that will tend to bind preferentially to particular ions or particular cofactors and so on. The composition of the peptides will depend on effectively templating from the RNA sequence, and as you get better and better at doing that, as proper selection takes over, and we're selecting now for the sequence of RNA, then we can be selecting for longer proteins and secondary, tertiary structures and so on. So I've not really thought through the steps yet. We're really focusing on the first steps, and do we now have an evolvable system when we have some form of templating of short peptides on a random RNA sequence? Will it evolve towards an optimal sequence? I mean, in my head it should. But modelling has a knack of confounding your expectations and making it, coming up with things that you just never thought of. That turn out to be obvious afterwards, but not at the beginning.
I think we have time for one last question, if there is one. Yeah, sure.
What do you think is, aside from, still not getting enough evidence, what do you think is the biggest criticism of your existing body of work?
I mean, I do think that evidence really is the biggest criticism. I think personally there's a lot of, if you like, suspended belief involved in any hypothesis for the origin of life. There's so many steps that need to happen, and you kind of think about this succession of steps, let's say to make ATP or something. That's a lot of steps. There's 11 steps in purine synthesis, starting from the precursors that I mentioned, which have got about 4 or 5 steps to get to them. So there's a lot that we're expecting of enzyme-free metabolism. And I think the biggest criticism, the most reasonable criticism is it's not going to happen. And they may be right, we'll try, we'll do our best. I think the other thing to realise is that the synthetic chemists who've done this kind of thing already have been doing it over decades. The Miller-Urey experiment was 1953. Oro was making adenine in the 1960s, starting with cyanide. People like John Sutherland here in Cambridge had succeeded in 2009 in making nucleoside phosphates, which was a big, big achievement. But we're talking decades after using cyanide to be able to make adenine. So, realistically, how long is this going to take us? How patient are people going to be? I'd like to think that we can crack it, but one thing I've learned along the way is that it's not always obvious which metal ion might be a good catalyst. I showed in uracil synthesis copper. I would say copper should not be very available in hydrothermal vents, in alkaline hydrothermal vents. It should precipitate out as copper sulfides. It wouldn't if there's a limited availability of sulfide. And we don't really know how much sulfide there was in these environments. I mean, there's plenty of scope for criticism, and there's plenty of need for some level of belief that it really can happen. Because otherwise, why do you get out of bed in the morning and try and do these experiments? And I don't want to ruin the careers of my PhD students. I want this to work. But maybe it won't. There's a risk. And so you hope for the best, and you try and find ways of doing other experiments that are more likely to work, perhaps in the meantime, that cushion out an idea and make you feel more comfortable with the overall thing. It's an exciting problem to work on. I don't imagine that we're going to really solve it. But I do feel as if in the last 5 years or so, I personally have gone from not really having a serious clue about most of the steps towards the genetic code to having an exciting hypothesis for how it might have happened that I don't quite find believable, but I certainly find it a lot more believable than well, anybody else's ideas.